At the leading edge of a migrating cell, just behind the protrusive lamellopodium, lies a broad flat region called the lamella, where the cell adheres to its underlying substrate. In contrast to the branched actin meshwork of the lamellopodium, the lamella contains several types of actin-based stress fibers. Jennifer Lippincott-Schwartz from the NIH in Bethesda, Maryland, and her postdoc Dylan Burnette, who was now running his own laboratory at Vanderbilt University School of Medicine in Nashville, were interested in how these actin structures are organized and how they might generate the flattened shape of the lamella.
So the general organization has been known for a long time. We have what are referred to as ventral stress fibers on the bottom of the cell, which are attached by adhesions at both ends. And we also have these actin arcs, which run parallel to the leading edge. And there are also dorsal stress fibers, which grow out of adhesions on the bottom of the cell, but then extend up to the dorsal surface.
However, due to the extreme flatness of a typical lamella, the 3D organization of these different actin structures is hard to resolve by conventional confocal microscopy.
So we decided to look at this in finer detail using structure illumination microscopy, where we could see things in 3D. And we saw that this system was just remarkably organized. All of the arcs that were forming off the edge of the cell were migrating up along the top surface of the cell instead of on the bottom surface. And as they migrate up along the top surface, they're maintaining an association with dorsal stress fibers.
Indeed, almost all dorsal stress fibers extend upwards from cell adhesions at the ventral face of the lamella to connect with actin arcs at the dorsal surface. In agreement with previous studies, the motor protein myosin 2A was largely absent from the dorsal stress fibers, but strongly enriched on the transverse actin arcs.
And what we were able to show using structure illumination microscopy is that the myosin 2A is organized as mini filaments in an identical fashion as the way they're found in sarcomeres.
With SIM, we can resolve the groups of head motor domains on both sides of the filament and also the tail domains, and so we could actually look at the structure of myosin 2. And so we originally saw that the myosin 2s were generally arranged parallel to the leading edge. And they grew over time, which is what we were really excited about, is that the myosin filaments typically first form as a single filament, and then over time they form stacks of filaments. And after these stacks are formed, they're actually separated by a major component of muscle cells, alpha-actinin, very similarly to a sarcomere. And many groups have proposed that the actin arcs are organized like a sarcomere, but this is the first time we've been able to actually show it.
This sarcomere-like arrangement allows the myosin 2 filaments to contract the actin arcs as they propagate backwards beneath the dorsal surface of the lamella. Burnette et al. Thought that this contractile meshwork might be able to flatten the lamella through its connections to dorsal stress fibers. Alpha-actinin localizes to both actin arcs and dorsal stress fibers, and speckle microscopy revealed that both types of structure move back from the leading edge at identical rates, suggesting that they might be mechanically coupled.
And we can put on blebstatin, which is a acidic inhibitor of myosin 2, and we see that the actin arcs and the dorsal stress fibers both stop. And because the dorsal stress fibers have no myosin 2 on them, the simplest explanation that we came up with was that as the actin arcs move away from the leading edge, they're actually pulling on the dorsal stress fiber, which then would pivot on the adhesion. And if they're pivoting on the adhesion, that makes the dorsal stress fiber a lever.
Pulling this lever down would, the researchers thought, cause the lamella to flatten. One prediction of this model is that rotational traction forces should be generated around the lever's hinge, that is, around the focal adhesions. And indeed, by measuring the three-dimensional displacement of fluorescent beads embedded in the underlying substrate, Burnette et al. Found that migrating cells pull their substrate upwards near the leading edge and push it down further back, behind the focal adhesions and dorsal stress fibers. Another prediction is that disrupting this contractile system should inhibit lamellar flattening. Burnette et al. Therefore knocked down myosin 2A, or treated cells with inhibitors of either myosin 2 or Rho kinase.
And so all three treatments remove the dorsal contractile network while maintaining a relatively normal-looking ventral stress fiber population, and the cell actually thickens. You go from having a lamella that's a few hundred nanometers tall to a micron.
COS7 cells lack myosin 2A and don't have a prominent flat lamella region near their leading edge.